I'm Dr. Leonard Gomella, Chairman of the Department of Urology at the Sidney Kimmel Cancer Center at Thomas Jefferson University in Philadelphia. And today we're going to be continuing the Grand Rounds in Urology discussion on genetics and prostate cancer. And specifically, the area that we're going to be talking about is identification of inherited prostate cancer risk. So there's a lot of information out there in the media about genetics and genomics, talking about DNA, talking about new gene tests. And as of a couple of years ago, there were over 60,000 genetic tests that were out there, either approved or being evaluated by the FDA. So this is a very, very hot topic in all of medicine, but in particular, prostate cancer over the last couple of years has been catching up in our understanding of how best to use genetics and genomics in the field of the management of the patient with prostate cancer. There's also things out there that I loosely call recreational genomics. These are things like 23andMe and Ancestry.com and other things that any person could go ahead and send in a buccal smear or a spit test and get information back on. And while these are interesting, I think that really our focus should be on tests that are really supervised and mentored by healthcare professionals to give patients the best guidance that we can with our current state of knowledge in prostate cancer. So why is this all happening? Why has there been such an explosion over the last couple of years? Well, this is mostly due to the government and the completion of the Human Genome Project. This project was funded by the National Institutes of Health and actually goes way back to 1990, where the goal was to sequence the entire human genome. And this is no small task. There are 3.2 billion base pairs in each cell in our body. Except for red blood cells. So you might imagine this was an enormous task that was started and took 13 years to complete. And obviously, early on in the process, the technology was not that advanced, where it took many days sometimes to sequence just several hundred base pairs, until later in the process with new advances in sequencing machines, much more efficient sequencing of the human genome was possible. So everything changed in 2013. And what do I mean by that? In 2013, Angelina Jolie brought the concept of genetic testing and cancer risk to the world through her discussion and her public review of her treatment of her BRCA1 and BRCA2 gene abnormalities that increased her risk of developing both breast and ovarian cancer. And of course, her very public discussion of this and her personal struggles with this to ultimately have prophylactic surgery performed introduced individuals to this concept of inherited genetic risk for developing cancer. Now, the focus, obviously, of her world was breast cancer. But obviously, as we're going to see, we've been following along now with prostate cancer, probably 3 to 4 years behind our colleagues in breast cancer when it comes to understanding the inherited risk of developing prostate cancer. And in fact, as we'll see over the next couple of minutes, it is the same genes, the BRCA1, BRCA2 genes, as an example, that increase breast cancer risk also appear to increase prostate cancer risk in men. So the things that we're going to talk about are basic concepts in this genomic and genetic testing world. We're going to talk specifically about prostate cancer and inherited risk. We're going to talk about some of the practical considerations in the clinical world of genetic testing. And lastly, we're going to talk about the very exciting and rapidly developing area of the emerging role of genetic testing in prostate cancer. So basic concepts, this sort of takes everybody back a little bit before we get into the deep discussion about prostate cancer, about in general, how are we using genomic and genetic applications? Well, you can see here from this small list, there's 3 general areas right now that we're using genomic testing: risk and screening assessment, the area of pharmacogenomics, and decision-making concerning treatment and adjuvant therapy. It's that last one, decision making in treatment and adjuvant therapy. I think that most of our urology colleagues are familiar with, because this is what's done with things such as the Decipher, the Prolaris, Oncotype DX, and those type of analysis, and they give you prognostic information. So that's where you actually look at the tissue, and it helps you in decision making. The second one on the list there is pharmacogenomics. We're not there yet in prostate cancer so much, but very often other drugs, such as drugs used to treat breast cancer and other diseases, rely on pharmacogenomics, where you look at a specific gene signature and then you match it up with a drug. We do have a little bit of this. It's not specific for prostate cancer, but pharmacogenomics for us using pembrolizumab for advanced prostate cancer. There's a very generic approach for this if you have so-called microsatellite instability in this particular, any tumor at all, including prostate cancer, patients are eligible to be treated with pembrolizumab. That's a very broad area, not really specific for prostate cancer, but that gives you some idea of the direction that we're going in, where you match up a patient's genomic profile with the treatment. Now, at the end of the talk, we're going to talk about where we're going in prostate cancer, matching drugs such as PARP inhibitors with genetic profiles, but these are not FDA approved yet. Lastly, risk and screening. Big area, particularly for breast cancer, very hot area. So genetics traditionally is the study of individual genes and their inheritance. It used to be very simple. You had a single gene abnormality that accounted for something like sickle cell anemia or something like cystic fibrosis. Today, this is much more complicated because many of our diseases involved multiple different genes, as we'll see, and the inheritance patterns can be quite complicated. The term genome refers to the entire set of genes in an individual organism. As we mentioned earlier, there are 3.2 billion base pairs in the DNA of an individual human cell. So when you use the term genome, you're talking about the entire set of genes in an organism. The word genomics is a little bit more complex, and when we use the term genomics, it really refers to the analysis of multiple genes interacting with each other and with the environment. Very important for diseases such as cancer and diabetes, where very often you cannot identify a single gene as being the cause of that particular cancer. So these terms loosely that we throw back and forth, when we use the term genetic testing, really today we're relying on genomic testing. We're referring to an analysis usually of multiple different genes in an individual to determine their risk of diseases such as prostate cancer. So a little bit more to bring everybody back to high school or college biology. We mentioned that the genome is an organism's complete DNA makeup. DNA is, of course, made up of base pairs, and as a relative ruler, bacterium have about 600,000 base pairs. Humans have greater than 3 billion base pairs in an individual cell. Of course, we have 23 chromosome pairs, and each has, all the chromosomes consist of 3.2 billion base pairs. So a gene, the official definition of a gene, is the sequence of these base pairs that actually make proteins. And in fact, humans are basically a bunch of proteins that are put together with a little bit of water thrown in. We have about 30 to 40,000 genes that code for proteins, and these are coded by parts of the DNA known as exons. These are the things that actually make DNA. However, this is actually less than 10% of our human genome. In fact, today, most of the DNA in our body is called non-coding introns. We don't know what a lot of them do, and certainly over the years we'll be discovering more and more about this, but the majority of our DNA is really non-coding. We actually don't know what its true function is. It could be structural, and some of these genes could be housekeeping. This is just a cute little graphic to understand the magnitude of what we're talking about. If the human genome was converted to a telephone book, it would comprise of about 200 telephone books with about 1,000 pages each. Compare that with something like a fruit fly, where the entire genome of a fruit fly might be in 10 telephone books, a yeast it might be in 1 telephone book, a bacteria such as E. coli may have a 300-page telephone book. The reason I point this out is because we are talking about concepts here that are beyond our human ability to get our arms around. The massive numbers of genes that we're talking about are something that we really need to rely on computers and computational biology to understand what is actually going on. And in fact, here's an example of why we need computers. So this is an example of just one portion of the BRCA2 gene. It's 27 exons. The coding region is over 10,000 base pairs, and this is just one small portion of exon 11. This BRCA2 gene is 12 pages long of AT, TC, GC, and so on. So I think you can appreciate the fact that trying to decipher this by the traditional Mendelian genetics that were popularized in the 1800s is just about impossible to do, and that's why we really need to rely on a lot of technology and computer strength when we start talking about genes and genetic alterations. So most of us are familiar with these karyotypes, and karyotypes are actually still used clinically. This karyotype here is an example of Down syndrome, trisomy 21. And here's the interesting thing is that major chromosome breaks, copies or translocations, you can actually see them under the microscope. But what we're going to be talking about in the rest of this lecture is the fact that most of the changes for cancer occur at the DNA level, and there's no way to detect these except using molecular biology techniques. So this is kind of my acute view overview of where we are with modern tumor evaluation. You know, we have great imaging. We can see the tumors now. Once we take them out, we have gross pathology. We can look at things under the microscope and see histology. We can even look at the cells and the nucleus. The problem is once we start getting down to the chromosomes and the DNA, you really can't usually see the cancer-related changes. So we have to rely on molecular testing, genomic analysis to understand really what's going on at the molecular level beyond the level of the cell and the nucleus to see why a cell, in our case today, prostate cancer, goes from normal to abnormal. So this is one of the key slides I hope that everyone will keep in mind when we start talking about this area of genetics and genomics. There are two concepts that everyone has to remember, and this is very important in urology. There are germline mutations and somatic mutations. Germline mutations are the abnormal genes that you inherit from your mother and your father, and possibly your grandparents and your great-grandparents. These so-called germline mutations are in every cell in our body. Very different than somatic mutations. Somatic mutations are things that happen in the tumor itself. And you can almost might think of it as the wild, wild west of the tumor. Some of these changes might be inherited, but most of them are spontaneous. They are just something that happens in the tumor itself, and it cannot be transmitted to any individual. So germline mutations can be transmitted to individuals, children, whereas somatic mutations are only the things that happen in the tumor itself and cannot be passed on to offspring. So what do we talk about now with clinical genetic testing? So in our world, genomic testing in urology has been common. It's performed in tissues, and you look at molecular signatures to help you with decision making. Decipher, Prolaris, Oncotype, Confirm are some of the more common commercial assays that we've used in urology for 5 or 6 years to help with decision making and management, active surveillance versus active treatment. There's another aspect to clinical genetic testing known as tumor sequencing, and this is where you go in and you actually get a piece of the tumor to look for somatic mutations. You send it to a laboratory and you get a complete genetic or genomic picture of the tumor. This is important today. Companies such as Foundation Medicine, Caris, and others look at these genetic abnormalities, and what they allow us to do is to specifically figure if there is a targeted therapy, a specific drug, a specific monoclonal antibody, or other agent that would be most effective in this cancer. So that's how clinical genetic testing is being used on the tissues. Our focus today is really on inherited cancer testing. These are the mutations that are inherited that don't necessarily cause cancer, but increase the lifetime risk. And in fact, as we'll see in prostate cancer, somewhere around 15% of prostate cancers can be identified as having some genetic or inherited basis. But the reality is 80% of prostate cancers today are sporadic, and we can't identify a specific genetic alteration. However, as we'll see, if you can identify a specific genetic alterations, it can make a big difference in how a patient who gets diagnosed with prostate cancer can be treated. So a little bit more about the basics of tissue testing. Again, everybody's familiar with Prolaris, where you look at a specific genetic signature in the tissue, done usually through archived, fresh frozen paraffin-embedded tissues. That is usually done through a pathology laboratory and then passed on to the commercial lab. And that helps with decisions about aggressiveness and prognosis. Germline genetic testing is most commonly done by sort of so-called spit tests or buccal smears, or occasionally by blood samples. And by doing that, you're identifying germline mutations that may be present, altered genes that may be inherited, because as we mentioned earlier, these genetically inherited genes are going to be in every cell of the body, and one of the most easy places to get a cell sample from is the inside of the mouth. Now, there's another concept to talk about as we finish up on the basics of genetic testing, and that's known as deep sequencing. Deep sequencing is very critical today to our understanding of genetic and genomic testing. Deep sequencing is sometimes called next generation sequencing. You might imagine that when you start getting down to this level of hundreds or thousands of genes with these ATGC sequences, that going in and analyzing that area one time, it may not be 100% accurate. So what deep sequencing does, it goes to a portion of the base pairs and goes back and studies that area and reads it over and over and over again. Why does it do that? It does that to limit errors. It does that to limit the chance that you might have missed a genetic alteration that could be important. Gene chip technology has really revolutionized this, where you put the sample and you put it in with the gene chip, and literally hundreds or thousands of genes can be analyzed from a relatively small sample. A cautionary here is that some of these so-called recreational tests, not all of them, but many of them don't necessarily do extensive deep sequencing and don't necessarily really look extensively on all the genes that may be abnormal. For example, as we're going to talk about mutated BRCA1 and BRCA2 genes, there are literally a thousand variations of how this gene can be mutated. So it's not just one or two. And this is where deep sequencing and going over the genetic makeup of a sample over and over again increases your accuracy. So hopefully that's given everybody a background so we can get into the area specifically about prostate cancer and inherited risk. So a basic concept is that all cancer is driven by genetic alterations, but we must understand that not all cancers are inherited. And in fact, that is the case of our prostate cancer population. As we mentioned earlier, maybe 15 to 20%, there can be some familial suggestion, but the majority of patients with prostate cancer today still have sporadic, non-inherited prostate cancers. So when we talk about cancers, we talk about three general cancers, approaches to how patients are diagnosed with cancer. We mentioned the sporadic cancers. The cause is not exactly known. You can't really see any features of other cancers running in the family. There doesn't appear to be an increased risk of close family members. And again, this is the majority of patients with prostate cancer. However, as we've mentioned several times, somewhere around 50 and possibly high as 20% of prostate cancer, there can be some familial suggestion that there is a genetic alteration in the family, in the germline, that increases the risk. Hereditary prostate cancer is a very specific entity where you can clearly identify the inheritance pattern that goes from grandfather to father to son. This inherited risk is very often due to something such as the HOXB13 gene that we'll talk about. This is very rare, but when you see it, it's very memorable. And this is one of those rare cases where this specific gene actually may be associated with the purely inherited prostate cancer risk. But most patients that we're able to identify some inherited component, there's familial clustering of cancers. And not all these cancers, as we'll point out, are prostate cancer. They may be some other related cancers. So one thing to be very clear about is when we use the term today in this lecture, BRCA1 or BRCA2, we are referring to mutated genes. We're not referring to the naturally occurring BRCA1 or BRCA2 gene. We're referring to mutations in those genes. And another important concept to stress is these genes do not cause prostate cancer. They do not cause prostate cancer. They do not cause breast cancer. However, when these cancers do develop, why are these genes important? They tend to make the cancers more aggressive. They tend to be more hard grade, and they tend to spread earlier than other cancers that don't have these particular genetic abnormalities. These genes, these inherited genes, also increase the risk in the individual and in the individual's immediate family of other cancers. Cancers such as breast, ovarian, pancreatic cancer. These increase the risk of male breast cancer. So again, they don't cause the cancer, but they increase the risk. So why do we do this genomic and genetic germline testing? Well, one of them is today, the hot area is the impact on therapeutic options. Doing germline testing and identifying certain inherited genes may direct you to treating a patient with a certain experimental drug that one day will likely be FDA approved and connected to a specific genetic test. But what we're learning about as well, and we're very early in the course of this genetic germline testing may be a way to alter our screening and prevent other at-risk cancers, not only in the individuals, but in their family members as well. So here you see some of the mutated genes that are commonly associated with prostate cancer: BRCA1, BRCA2, ATM, CHEK2, and others. But what's very interesting is these genes, almost all of them have something to do with DNA repair or DNA damage response. So these genes have a very common familial origin in how they're actually interacting in a cell to cause increased prostate cancer risk. I mentioned earlier this HOXB13 gene is one of the genes that is most clearly associated with inherited prostate cancer. Multiple male members of a family who develop high-grade cancer at a young age. Very often the gene, the HOXB13 gene, as you can see, which is an androgen receptor repressor, if it's mutated, may increase the risk of prostate cancer in an individual. So this was one of the most famous papers by Dr. Colin Pritchard at the University of Washington in Seattle. It was published in New England Journal of Medicine. When you looked at men with metastatic prostate cancer, you can see that there were germline mutations in about 12% of those men. Most of them were BRCA1, 2, CHEK2, and ATM, but there were many other mutated genes. And you can see that these are germline mutations. This means that these were inherited abnormal genes in these individuals that increase their risk of developing metastatic aggressive prostate cancer. So BRCA1, BRCA2 mutations in prostate cancer. These are DNA damage response genes. When they are altered, it interferes with the cell's ability to undergo proper DNA damage repair, leading ultimately to uncontrolled growth of the cell. They increase the risk of prostate cancer. BRCA2 appears to be more critical in increasing prostate cancer risk than the BRCA1. If you've got any of these gene abnormalities, your prostate cancer is likely to be more aggressive, likely to be a Gleason 8 or higher, more likely to have positive nodes, and in turn have poor survival. If you've got these mutated genes that not only increase the risk in you, but also in your family members, if they also inherited these genes for other hereditary cancer syndromes, such as breast, ovarian, melanoma, pancreatic cancer, Lynch syndrome leading to increased colon cancer and gastric cancer. But as I mentioned earlier, these germline mutations may, in the near future, direct how we treat metastatic castrate-resistant prostate cancer with a new class of drug known as PARP inhibitors. And we'll talk about that in a couple of minutes. So here's an example of the bad things that can happen if you develop prostate cancer and you have one of these germline mutations. You can see here from this paper from Hopkins a couple of years ago, a very significant difference in death rate from prostate cancer. You can see that the men with the germline mutations have a median survival of 11 years, whereas men with prostate cancer who don't have these altered genes have a median survival of almost 18 years. So again, very clear that these altered genes do impact survival and impact the aggressiveness of cancer, but again, don't necessarily cause prostate cancer. So there's no question that when you look at genes such as the mutated BRCA1 or 2 in breast cancer, the impact is clearly much higher. So this area is much better studied in women in breast cancer than men with prostate cancer. But you can also see that men who do have this have an increased risk of developing male breast cancer, as also the increased risk of developing prostate cancer, melanoma, and pancreatic cancer. So again, this is just something to keep in the back of your mind when you're evaluating patients, particularly those men with high-grade or aggressive cancers, that you may need to think a little bit more broadly about their overall health and cancer screening. So we keep mentioning that since these are inherited genes, these inherited germline mutations also increase the risk in other members of the family with this gene. So again, for men, it increases the breast cancer risk by 68% over the general population, increases prostate cancer, pancreatic cancer risk, and so on. You can see that the risk of having cancers such as ovarian, breast, and uterine cancer are very significantly increased in female members of the family. So these are hereditary familial cancer syndromes. So today we not only need to think about our man with prostate cancer, but we need to think about his larger family. So when we get down to nuts and bolts, what proportion of patients with localized prostate cancer have germline mutations? Well, if you look at BRCA1 or 2 for localized cancer, it's actually quite low. And you say, okay, that's fine. But here's the interesting part. If you've got someone with a localized prostate cancer, but they have this germline mutation, they act more like patients who have metastatic disease. In other words, these patients genotypically end up looking like metastatic castrate-resistant prostate cancer, and even though they've had no ADT at all. So again, clearly, limited numbers of men with localized cancer have these abnormalities, but they turn out to be bad actors. Next, let's talk about some of the practical considerations in genetic testing. We were very fortunate in 2017 to have the first international consensus conference where we brought together a large number of individuals with varied backgrounds to talk about this rapidly evolving area of genetic testing for inherited prostate cancer risk. The paper was printed in the Journal of Clinical Oncology back in February 2018. And these were some of the things that we addressed in the consensus: which men should undergo genetic testing and counseling, which are the genes we should be looking at, and what genes should be factored into management concerning screening, early prostate cancer, advanced prostate cancer. So what's happened now in the field of medicine is that this area over the last 2 or 3 years of genetic testing in men with prostate cancer is getting the proper amount of attention that it deserves. I'm showing you this 2018 NCCN guideline, but this is not a prostate cancer guideline. This is for HBOC, hereditary breast and ovarian cancer syndrome. You actually got more information before 2018 on how to manage men with these genetic alterations by reading the breast cancer literature as opposed to looking at the prostate cancer literature, because the researchers in the field of breast cancer recognized a long time ago the familial risk of this is very important in men and women if they have a BRCA abnormality. So in 2017 was the first time that the NCCN noted the BRCA when it came to prostate cancer early detection, and basically suggested that if a patient has a familial or personal history of a BRCA 1 or 2 mutated gene, that should be factored into when you begin screening. So again, this is not hard and fast yet, but at least we're now starting to think about men with genetic alterations and how that should impact their screening. In 2018, the NCCN came out with the first table that actually recommended when germline testing should be done. And this is a very useful table, and I recommend that everybody have this or the updated version of this from NCCN in their clinic to help guide them on which risk groups of prostate cancer should have genetic testing. So practically speaking, commercial laboratories have a series of prostate cancer specific panels. You see a listing of some of them here from a variety of commercial laboratories. While all of them tend to have ATM and BRCA 1 or 2 and check, some of them have very different genes in there. And again, it's important for you as a clinician working with a genetic counselor to understand that not all prostate cancer specific panels are the same. Other common cancer panels are such as the one from Myriad, known as MyRisk 28 Gene Screen, that actually looks for a variety of cancers. It doesn't focus just on prostate cancer, but tries to identify the common inherited familial genes that are associated with breast, ovarian, colorectal, and the like. Our commercial colleagues are very helpful. As urologists, we tend not to be good with family histories, and our commercial colleagues have developed these very useful questionnaires that I think that urologists should seriously think about incorporating into their intake, particularly for men with high-grade or aggressive prostate cancer, should really be given the opportunity to give a good genetic familial history, because it may impact not only the individual, but members of their family. So here at the Jefferson and the Kimmel Cancer Center, we started a multidisciplinary clinic way back in 1996 for all the patients to come in with newly diagnosed prostate cancer to see radiation oncology, medical oncology, and clinical trials coordinators. In 2014, we developed, we believe to be the first genetics clinic where all men coming in with prostate cancer have the opportunity to be screened and evaluated for genetic abnormalities. We do this with our outstanding genetic counselors, Dr. Vita Giri and others. We have research studies going on, us and other individuals around the United States, trying to get a better understanding of exactly how should we be using genetic testing to guide prostate cancer treatment and diagnosis. I put a little plug in here for genetic counseling. I talk a lot with individuals about genetic testing, but I think as surgeons, as urologists, we really need to partner with genetic counselors because this is a very complicated and rapidly evolving field. And while we need to have an understanding about the genetic test, I actually think when it comes down to what test really should be done, it's based on the family history. And I think this poor woman indicated in the slide here really never saw a genetic counselor. Everybody make assumptions that she had a certain hereditary component, and again, did not give her the best care possible. So I think genetic counseling for inherited prostate cancer risk goes beyond the genetic testing. The genetic counselor will look at things such as family history, personal cancer features, and other risk factors, and then come up with a recommendation not only for the individual, but also for the possibility of other members of their family. And very often, patients that we send for genetic testing don't really have any signal that they have inherited prostate cancer risk. Again, because remember, 20% or less of our patients actually had this, 80% are sporadic. Nonetheless, when you identify it, you need to allow the patient to make an informed decision how much genetic testing they want to undergo and so-called cascade testing of other members of the family. So here's an example of a family tree. And as a urologist, when I look at this and I look at histories of prostate cancer and histories of colon cancer and people who died of their diseases, it's a bit overwhelming to me. And I think this is the kind of patient that really would benefit from talking to a genetic counselor and determining if other members of their family should undergo so-called cascade testing. One last little point just to mention is something known as the VUS, genetic variant of uncertain significance. This is something that urologists who are ordering genetic testing need to understand. It basically means that some funny genes were identified, some funny mutations in key genes were identified, but we don't know what it means. Why is this VUS important? When you go through a genetic counselor and the genetic laboratory identifies a VUS, it goes into a large database. That database gets looked at over and over and over again. And very often, 6 months to a year later, the database says, you want to know what? This is just a random change in the gene. There's no increased association with a specific cancer. However, there are VUSs that do come up, and you can see here example of BRCA1, BRCA2, EpCAM genes, and things like that. And you can see that many of those genes do have a VUS rate that actually gets classified at some point as being an important malignant gene. Lastly, some of the practical issues is known as Gina, Genetic Information Non-Discrimination Act. If you get involved with genetic testing, you need to be familiar with Gina. Why is that? Because it does not protect patients against discrimination in life, disability, or long-term insurance care. It does protect them with health insurance. It does protect them in employment, but it's very important that you let patients know that once they have a genetic test and if they are identified with a gene, it may have impact concerning things such as life or disability insurance. Question always comes up about insurance coverage, and again, it's all over the place. One thing we can say is that Medicare is very good. If a patient has metastatic prostate cancer or they have a very strong family history of a hereditary cancer syndrome, Medicare will usually cover genetic testing and counseling. Other insurance companies, it's very, very highly variable. So lastly, where are we going in this area? What's the emerging roles of this? Again, in 2019, now there has been increased emphasis placed upon familial or personal history of germline mutations when it comes to early detection. These are the latest NCCN guidelines that put increased attention on this susceptibility of mutated BRCA1 or 2 genes in particular. Again, germline testing in the 2019 update is much more heavily recommended as opposed to being suggested. If you remember in the earlier 2018 guidelines, a lot of suggestions were made or to be considered. Now you get much, much more hardcore recommendations about germline testing based on the clinical risk group. Genomics connect very well to precision medicine. A very exciting development was published by Bal Carter and Associates late last year, looking at active surveillance, noting that if men had an altered BRCA1, 2, or ATM, that they were more likely to be upstaged or upgraded on active surveillance. Perhaps, again, not doctoring yet, but perhaps in the near future, we'll be doing genetic testing as a way of discriminating men who have the best risk for staying on long-term active surveillance. Here you see clinical studies that are being done in men with advanced prostate cancer, identifying the best approach to patients based on their BRCA1, BRCA2 status, showing certain patients work better with BRCA2 mutations, comparing enzalutamide to the taxanes. You can see that if you're BRCA2 positive in some of these studies, you don't do very well on taxanes, but you do better on some first-line treatments such as abiraterone or enzalutamide. One of the most exciting areas is going to be the availability of a new class of drug called PARP inhibitors. These are already available for cancers such as ovarian cancer. Studies now are being completed where if men have a DNA repair response alteration, i.e. a BRCA1 or 2 abnormality, they are more likely to respond to PARP inhibitors, in this particular case, olaparib, and basically these drugs have been granted breakthrough therapy. So germline testing may actually be important in men with advanced prostate cancer when it comes to deciding the treatment. So what's the bottom line on genetic testing and counseling for prostate cancer? Well, if you have a patient who has more than 2 cases of prostate cancer at young age and close relatives, more than 3 first-degree relatives with prostate cancer, aggressive prostate cancer associated with breast or ovarian or pancreatic cancer in a close relative, all metastatic prostate cancer, all intraductal prostate cancer, and again, tumor sequencing should be considered in addition to germline testing to look for local somatic alterations that may be treatable. So as I mentioned earlier, we had a consensus in 2017. We have a consensus now planned in 2019 here in Philadelphia, where we're going to look at the actual implementation of genetic testing and trying to get a better handle on exactly when and how to do this genetic testing and counseling. So the bottom line is, in urology, we've been well established using prostate cancer genomic tissue testing through tests such as Prolaris and others. We now have rapidly evolving recommendations for prostate cancer genetic testing and decision making. Again, the most critical inherited genes there are listed. We know that if you've got metastatic prostate cancer, we really want to know if you have a germline mutation because it may direct how you're treated. And I think referring men for genetic testing and counseling, particularly if they have high-risk personal features or there's a family trend of an inherited potential mutated gene, I think it's very important. I think we need more and more genetic counselors to work with us in the field of urology. This is a rapidly expanding field, and we know we don't have enough genetic counselors out out there, but we need to work closely with those that are available to help us care for our patients in the best way possible. So I thank everybody for that, for their attention. I presented you with a lot of information in this lecture, but I hope that we hit the highlights so you can effectively begin to understand how to incorporate genetic and genomic testing in your practice. Thank you, and I thank the team at Grand Rounds in Urology for providing me with this opportunity.